My name is Christine Leb. I'm working at Vienna in Austria at the University of Natural Resources and Life Sciences, and I'm very much looking forward to the talks. However, before we begin, but are they now still here with us? Vlatko, there was supposed to be an announcement of today's workshops in the afternoon. No, they are not here anymore. Maybe we'll do that straight after the talks. So stay with us because they want to explain maybe also how the workshops are going to work. So yes, just now to start with the first presentation, I would like to invite Michelle Reeves to come forward and to present our work on the impact of gastrointestinal parasitism on lamb behavior and welfare. Please.
Thank you very much. Hello, everyone. My name is Michelle Reeves from SRUC, and today I'll be talking to you about the impact of gastrointestinal parasitism on lamb behavior and welfare. So gastrointestinal parasitism is an important health and production challenge that sheep in the UK that graze on pasture face every year. Lambs are especially susceptible since they haven't developed any immunity to the worms yet. And we know that it can cause important health problems like abomasal lesions in the gut, and it can have a negative impact on production through reduced feed intake, which can lead to weight loss. What we know a lot less about is the welfare impact of these parasite infections. And this is despite the fact that a 2020 paper by Rioja Lang et al. Found that UK experts consistently ranked parasitism very highly in a list of welfare challenges faced by sheep. And these experts were particularly concerned about the high prevalence of parasitism in the UK. And these results were echoed in a stakeholder meeting, which included farmers, that was held by TechCare, which is the EU project that funds my PhD, where actually the UK stakeholders ranked parasitism as the #1 concern faced by sheep. So how do we get a glimpse into the experience of a lamb that is infected with a parasite? Well, we have some behavioral indicators that can give us some hints. For example, parasite-induced anorexia, changes in diet selection, and changes in grazing behavior. But what do these changes in behavior mean for the animal's experience and affective states? If we imagine a graph with day of infection on the x-axis and parasite burden on the y-axis, how does the animal feel here at the onset of infection? And is it any different from here near the peak of the infection or here after treatment? If lambs are experiencing negative emotions or negative experience during parasitism and changing their behavior accordingly at the onset of the infection, maybe we could use those behavioral changes as very early indicators of parasitism. So these gaps in knowledge led us to ask the following questions. Do sheep experience negative welfare states in early subclinical infection? And if so, when do these set in? And can we use behavior and behavioral change to detect the onset of parasitism and this possible negative welfare state that comes with it? So to answer these questions, we ran a pilot study in the first year of my PhD, and we used both quantitative and qualitative methods in the study. Our first hypothesis was that lambs subclinically infected with parasites would be more likely to be lying and less likely to be eating and walking than uninfected lambs. And secondly, that infected lambs would score more highly along qualitative descriptors such as listless, physically uncomfortable, and apathetic, and score lower on terms such as social and content than uninfected lambs. And both of these hypotheses were based on the idea that infected lambs would be lower energy, less social, and less active. So we had 96 Suffolk crossed lambs in groups of 4 across 24 pens, and each block of pens was assigned to one treatment group. We had an ad-lib fed control group in pink, a restricted fed control group in orange, and a parasitized group in blue, which was trickle infected with the nematode Telodorsagia circumcincta. Our restricted fed control group was fed the same amount that the parasitized group voluntarily ate, and that was to separate the possible welfare effects of parasite-induced anorexia from the possible effects of the actual parasite infection in the gut. We also set up cameras over 12 of our pens to monitor the animal's behavior. And with those cameras, we took 1-hour clips every day at the same time of day for 26 days, and we analyzed those clips using 3 scan samples at 30-minute intervals. We also conducted qualitative behavioral assessment, or QBA, as developed by Françoise Wemmelsfelder at SRUC. These were weekly observations, always conducted by the same observer, which was myself. They were pen level observations as opposed to individual observations. So each pen was observed and scored on a list of 21 terms, terms that included things like alert, active, relaxed. And this list of terms was previously validated for use in sheep by the AWIN project. The data was collected on an app for QBA that was developed by SRUC. So this table shows you the amount of times that each behavior was recorded in our scan samples. As you can see, lying was a very prevalent behavior, and there was no difference across our treatment groups in the probability of lying behavior. And this is likely because the animals were indoors, and this affected their behavior, because when you look at time budget studies of lambs of a similar age that are outdoors, they tend to spend a lot of their time, 50 to 75% standing. We did look into standing and eating behavior as well. However, we couldn't analyze the data from the other behaviors because they were recorded very rarely and therefore had very little variation in the data. So we modeled standing behavior using generalized linear mixed models, where standing was a binary, and the variables we included were day of infection, which is DOI here, treatment group, and scan number, as well as the interactions between each of those variables. And our random effect was individual ID nested within pen number. And what we found was a significant interaction between day of infection and treatment group. In the parasitized group, there was a marginal increase in standing behavior over day of infection, and that was significantly different from the ad-lib-fed control group. There's a possibility that this might indicate an increase in discomfort behavior during the onset of our infection. So this graph shows those results. The differences between the treatment groups are not very obvious at first sight, but I'll walk you through it. We have day of infection on the x-axis and probability of standing behavior on the y-axis. In our pink and orange treatment groups, which are our control groups, we have a slight decrease in standing behavior over time. And in our blue parasitized group, there's a marginal increase, the opposite relationship, over time. We modeled eating behavior in the same way, and we found a significant interaction between scan number and treatment group, where the parasitized group showed a marginal decrease in eating behavior between scan 1 and scan 2, as well as between scan 1 and scan 3. That was significantly different from our control groups. So this graph shows the possible impact of parasitism on eating behavior. We have our scan numbers on the x-axis and probability of eating on the y-axis. As you can see, the scale of the y-axis is quite small, so these are very marginal changes. And the star numbers that match next to the dots show significant differences. So in our pink ad-lib-fed control group, we have a significant decrease in eating behavior between scan 1 and scan 2, but no other significant differences. In a restricted fed control group, we have no differences across the entire sample period. However, in our parasitized blue group, we have a significant decrease from scan 1 to scan 2, and that remains from scan 1 to scan 3. So scan 1 was the time that was closest to when the animals were being fed at the beginning of the day. And it's possible that the eating behavior remained relatively steady throughout the sample period for our control groups, whereas after that first scan sample, it remained low in our parasitized group. Our qualitative behavior assessment data was analyzed using principal component analysis, or PCA, and the PCA found two factors that accounted for a lot of the variation in the data. The first factor described activity levels, or rather calmness, went from active to calm, and accounted for 35.9% of the variation in the data. And the second one described the valence of the animal's affective states, going from listless and apathetic to alert and bright, and described 14.2% of the variation. So each of our pens were placed somewhere in the four quarters of this graph, and there was a graph for every week of the trial, 1, 2, and 3, where week 1 was prior to the infection and weeks 2 and 3 were during the infection. And these were our results. You can see the week changing in the top left. And as the ellipses all overlap for our 3 treatment groups, we saw no significant difference in the study of the placement on these 2 axes. What you do see is a change in the variation or the spread of the data. For example, you can see our blue parasitized ellipse really contracting during week 3, so the spread of the data is reduced. Things to consider about these results are that the indoor environment of this study definitely had an impact on the lamb's behavior. We're working with a relatively small number of pens and a small number of observations in animals. And as we saw with those small scales, the changes were very minor and marginal. So technology could be a very interesting tool to apply in these kinds of situations where maybe technological tools could detect very subtle changes that my in-person observations could not detect. So if we go back to our original hypotheses to conclude, we thought that our infected lambs were going to be more likely to be lying due to low activity, but in fact, they were more likely to be standing, possibly due to an increase in discomfort behavior. We did find that there was a decrease in the likelihood of them eating over our scan sample period, and we were not able to look at walking or locomotion behavior due to low variation in that data. In terms of our qualitative analysis, we didn't find any differences between our treatment groups in this study. Our next steps are ongoing. We're currently doing a large-scale outdoor trial that is a scaled-up version of this pilot study, where we're looking at more sheep over a longer period of time, and also applying the use of technology to detect these possible subtle changes in behavior and try to understand what they can tell us about the animal's welfare. Thank you very much for your attention, and I'll gladly take any questions.
Thank you very much for this lovely talk and being perfectly on time. Who wants to start to ask questions? I see already one. Where's the microphone, please? Yes. Please remember to introduce yourself briefly.
Sabine Gebhardt from the University of Bern. Thank you for your talk. What did you mean by subclinically? Because I mean, if you expect changes, then obviously it would not be subclinical infection rate. And so was this based on some other symptoms? And was there work before that you knew that this level of infection would not induce your symptoms that you were expecting? And which symptoms are those?
Yes, so that's a good question. So we were using doses that had previously been shown to the trickle infection. We used doses that were previously shown to cause subclinical infection. And we were actually particularly interested in the period before any symptoms are very clear or obvious, because the ultimate aim of our research is to detect disease and welfare concerns before the clinical signs are obvious, to really try to be as early as possible in our treatment. And so we were purposefully looking for small changes before any of the usual symptoms are visible.
Okay, thank you.
Thank you. There's another question. Just go straight over, yeah.
Really nice talk, Heather. This is Suzanne Millman, Iowa State University, and I'm delighted to see more work in the sickness behavior field. My question may be a little bit similar. I was trying to understand if point number one was before you infected them or if you had baseline, because one of the challenges with sickness behavior is knowing that animals were healthy coming in and they didn't have a concomitant disease or something else. So I just wondered if you had been able to control for that.
Yes, absolutely. So the animals were fecal sampled prior to first infection for 2 weeks and throughout the entire infection or the entire experiment. And so we did know that their fecal egg levels were 0 for all treatment groups at the start, and the control groups remained at 0 throughout the experiment, while our parasitized group did see an increase in its fecal egg count, but never went above subclinical levels of around 600 eggs per gram.
Thank you very much. There's no online question as far as I can tell. Any other question here? Yes, please.
François Martin, Nestle Purina. Thank you for the talk. Very interesting. You say that the next step is to use technology to dig deeper into this. Do you have an idea of what you will do, or is it? Can you share?
Yeah, absolutely.
Okay.
I'm happy to share. So we have lambs in the field. So this is an outside trial in very normal commercial lowland conditions for the UK. and they're wearing collars that contain accelerometers and Bluetooth proximity loggers to look at social interactions and distance between individuals. And so we're hoping to dig for any differences in activity levels, activity intensity, and social behavior as welfare challenges such as lameness, mastitis, and parasitism set in.
Thank you.
Thank you very much.
Thank you very much for this wonderful and very interesting talk. And you want to change, you can change now the room. However, I would recommend to follow up because we have a very similar topic on the same, also on sheep and parasites. And I would like to introduce Lena Lidfors from Sweden. And she will present her talk on influence of weaning age and natural acquired nematode infections on the behavior of lambs. So please, Lena, welcome.
Thank you very much. So this study has been mainly done by Niklas Högberg, who was a PhD student in this study, and Anna Hessle, who is an agronomist and know the fields and manage the research station where we've done the study. And Johan Höglund, who is a professor in parasitology. Let's see. So gastrointestinal parasites is a welfare problem for lambs, as you have just heard. And often they have a mixed species infection when they are out grazing. And there are several clinical signs of these infections like watery diarrhea, dull hair, coat, anorexia, and loss of general body condition. Additionally, if the lambs have Haemonchus consortus infection, they can have anemia and submandibular edema. And it's quite often found that the weight gain is affected negatively by the infections. And you can see a figure here of how they can be infected by the larvae on the fields. So the aim of this study was to investigate how behavior and body weight gain in naive grazing lambs were influenced by weaning age when exposed to contrasting levels of gastrointestinal nematodes under natural grazing conditions. So the predictions were that we would have a lower weight gain in the lambs with high parasite load and more lying and less grazing and ruminating in lambs with high parasite load. The material is that we did the study on our research farm in the southwest of Sweden. We had 41 multiparous Dorset ewes with twin lambs that we borrowed from a commercial farm. So the ewes were sent back to the farmer when we had weaned the lambs. They were released into 2 permanent pastures, naturally contaminated by sheep with strong guile nematode larvae the previous years, it should say, because we have had sheep for several years there. So we split the lambs up into different parasite load, the ones with the low parasite, and ewes and lambs were dewormed before turnout plus at weaning with ivermectin. And then we had high parasite load where we didn't treat either ewes or lambs. And then we also had a different weaning age. So half of the lambs were weaned at 10 weeks and the other half was weaned at 14 weeks. So you can see here a number of lambs in the different groups. We only had one group per weaning and parasite load, so we don't have multiple pastures with this. So the study design you can see here. So they were first kept in semi-natural pasture, and then after weaning they were kept on lay pasture. And we put on ice cubes recording on the hind leg of all the lambs that were at the weaning, and the drenching was done for those with a low parasite load at the same time. And then also Niklas was collecting fecal samples, and we were weighing the lambs. So all the feces was analyzed in the lab in Uppsala. And on the lower axis, you can see the days on pasture, when different things happened. So the behavioral observations were done by me, and I observed all the lambs in the 4 different groups at week 1, 2, 3, and 4 after weaning, and we put out separate days there. And the lambs were individually numbered with lamp spray on their backs, so I could identify the different individuals. And then each group had a different color. And they were observed in the morning, and then I switched between the groups who was recorded the first in the morning. The recordings were made instantaneously at every second minute at individual level for 60 minutes. And also the ice cubes were recording the lying, standing, number of lying bouts and motion index. We have previously validated the ice cubes and ice tags for growing lamps, and we found out that the number of steps is not correct when you put ice cubes on lamps. So we skipped that completely. And the analysis, the eggs were, the fecal egg samples were analyzed in the lab, as I said, and they also analyzed the proportion of different parasites, but I'm not going to go into that here. You can discuss this with Niklas if you have questions. No lambs really got sick during the 4 weeks after weaning, but I could see some lambs that they had like feces on their behind, so maybe some signs that they had mild diarrhea. I used a generalized mixed model to test if there were any behavioral effects of treatment, weeks, and interactions between treatment and weeks. So first, the results on the fecal egg counts. The average fecal egg counts was higher in the early weaned high parasite load lambs than in all the other groups. So there were no significant differences in the late weaned groups between high and low parasite load. And you can see on the upper figure, I don't know if I have a pointer here. Does it work? Up here on the higher figure, you have the fecal egg counts for the different times, and then you have the cumulative egg counts in the lower. And then you can see how much higher it was for the high parasite load lambs here on these two figures. And this is to give you the background that actually the early weaned high parasite load lambs were the ones who were most affected by the parasites. And if we then look at the body weight gain, we had significant differences between the high and low parasite load lambs when they were early weaned, but no differences for the late weaned lambs. And it was 11% higher body weight gain in the early weaned low parasite load lambs. So here you have a figure of that. If we look at the ice cube data, the average motion index was 19% lower in the early weaned high parasite load compared to the early weaned low parasite load, and it's on the upper axis. You can see the motion index. Here you have the lying behavior, which I was most interested in, and the average 24-hour lying time was 15% shorter in early weaned high parasite load. So they were lying less when they had a lot of parasites when they were early weaned compared to the low parasite load. There were no significant differences in the number of lying boats. Here are the direct behavioral observations. And here we also looked at lying, but I would say that I think the ice cube data tells you much more about the lying behavior, and it doesn't really correlate very well with the ice cube data, because here the lying was mostly observed in the late weaned high parasite lambs. If we looked at the grazing and ruminating, we can see that the early weaned low parasite load lambs were grazing much more than the early weaned high parasite load. There were not so big differences between the those which were late weaned. Oppositely, we see that rumination is less in the early weaned low parasite than the early weaned high parasite load. But it's the opposite picture when you look at the late weaned lambs, where you have more ruminating in those with a low parasite load. So it's actually an interaction here also between the weeks. So I think this figure tells a bit more what's happening in their behavior. So here I have put grazing and browsing together, and here you can see that, let's see if I can, the early weaned with low parasites were grazing a bit more here than the early weaned with high parasite load, but then they come closer together in the second and third week, but then they drop in the fourth week, those with high parasites. And if you look with late weaned lambs, we have the black gray line here. They have high parasite, and those yellow are the ones with low parasite load. So you have an increase here in the grazing over time. And I think we need actually this PLF monitoring equipment to have correct and good data on how much they are grazing. And also with the rumination, it would be nice to have more techniques. But here you can see also with the early weaned low parasites that they had less rumination compared to the early weaned with high parasites. And also for the late weaned, those with low parasites are a bit higher compared to those with high parasites. Then I did a more detailed analysis of each behavior, because here I had to group together all the ruminating behavior, both standing, lying, and walking. So if we look at the lying and ruminating, then I could see with the Kroskal-Wallis test that actually both groups of high parasite lambs were lying down and ruminating more than those with low parasites. We also see standing and ruminating a bit more in those who are late weaned. But if you compare within the early weaned, those with high parasites still have a bit more standing and ruminating. Also, the standing behavior is performed more in the high parasite load, as you heard from the previous presentation, compared to the with low parasites. But sometimes it's difficult to compare the early and late weaned lambs, so we may need to separate them better. Concerning the walking, there were more walking in the early weaned lambs. Yes, and one thing that I noted was that the early weaned high parasite load lambs, they were eating minerals more and drinking water more, but it's so low numbers. So I think it would be nice to be able to measure the amount of water and how often they go and drink water, and also how often they go and eat minerals. So maybe they're trying to compensate for some feelings in their body that they have parasites, but it's subclinical. Also concerning social interactions, we had more social interactions in those with low parasite load, both if you compare within the early weaning and late weaning. And there are no differences in lying still or grooming. So in conclusion, early weaned lambs with high parasite load compared to low parasite load have higher fecal egg count, lower body weight gain, lower motion index per 24 hours over all the 4 weeks, shorter lying time per 24 hours over all the 4 weeks, and lower percent grazing and browsing and high percent ruminating. However, there were no significant differences between the late weaned high parasite and late weaned low parasite. So lambs with high parasite load were more affected by early weaning than lambs with low parasite load, which might be interesting to consider when you discuss weaning age of lambs. And here on the picture, you can see the group with high parasite load, the early weaned compared with the other group. So thank you for attending this talk. And this study was funded by several different, the Research Council and other councils. Thank you very much.
Thank you very much for this very interesting talk on an important topic. Who wants to ask? Yes, straight away, your colleague.
Yeah.
Thank you, Michelle Reeves, SREC.
Yeah.
I have 2 quick questions. When you were doing your behavioral observations, were you blind to the treatment groups of the animals?
I tried to be blind to it, but I can't lie and say that I was totally blind, because they had colored collars and so on.
So the collars were color coded?
Yes.
Yeah.
Mm.
Okay. And did you have a treatment threshold, like a treatment criteria for the high parasite lambs, that if they reached a certain level of infection, they would get treated, or did you let the infection take its course for the 4 weeks?
We let the infection take its course, but if they would be really sick, like not lying a lot, or that you could see signs that they were sick, then they would be treated. So there were personnel on this research farm that went out daily to walk through the pastures and check that there were no sick lambs.
Thank you.
Thank you. One also from here, from the audience, just in the 3rd row, please. There's no online, so we stay here in the room.
Thank you. Lucy Oldham, SIUC. That's a really interesting thing to try and measure prior to infection. I'm interested in the social behavior. What did you count as a social interaction?
So that would be sniffing, mounting. They did some headbutting when they back off and bump their heads. These were all male lambs. So that I think that was the main social interactions that I saw between them. I could split them up because I have also, I didn't present now, but I have the frequency of vocalizations and each social behavior, and also looking for abnormal behaviors.
That's great. So you might see differences between different types when you get further into splitting it up.
Yeah, I can show that data. Yeah.
Thank you.
Thank you. One last question possible? No, not really. Otherwise, just from my side, briefly, what do you think about what is the starting level of infection? Because it's not always zero, isn't it? And from how early onwards to detect signs? What did you also look at this other threshold, not the upper, but the lower threshold?
I'm not an expert in parasites, so I think Niklas, he's a veterinarian, and he can probably talk more about that.
Yeah, I think just from I think it's very interesting to discuss also where does it start an animal to be ill or not ill? Yeah.
I could say that he has defended his PhD thesis, so it's available online at our university as a PDF file. Yeah.
Thank you very much. I think that's for all of us further reading. Thank you very much. So. Again, time to change if you want, but I'm sure you all stay here because we have another talk on this topic. The third talk is coming from Serbia and it's online. And I'd like to introduce now Katarina Nenanovic. She will talk about welfare and parasitology conditions of extensively reared sheep during winter. And I think she's here already and she can hear us, and maybe you're already sharing your presentations. Thank you for that.
Hello, can you see my presentation or?
Just put it on the presentation modus. We can see it already.
You can see it.
Yes, but if you click on the, what is it, on the slideshow modus on the bottom, what is it on from your side, right?
Like
presentation
this?
Modus, yes, yeah, here.
So it's okay now, or because I can't see
it doesn't change, but maybe it takes a bit. Press once more, maybe.
I don't know what to do, really.
No, otherwise we just look at it like that, but it's a bit of a pity if you just go away maybe once more with your
maybe
cursor.
Like this, I don't know. Maybe
or go to view and then say slideshow. So on the top to say slideshow. If you go very up with the cursor to the presentation, to the top of your presentation, where it says slideshow.
Okay,
or view.
Slideshow, but write your screen share this house.
Doesn't work. Do you have two screens? Maybe that's the problem. Maybe no.
No.
Otherwise, you just present it from here. Just click from one slide to the next. No problem.
But it is not perfect than the presentation, but I will try to present like this.
I'm sure it's fine. So,
my name is Katarina Nenadovic, and I'm associate professor from Faculty of Veterinary Medicine, University of Belgrade. And I can say only it's a great pleasure to be here. And two presentations before me was really interesting. And like I see, this parasitology condition of sheep is a very topic theme today. And like you see in my presentation, I will speak about welfare and parasitology condition extensively reared sheep during winter in Serbia. And like we all know, firstly in Serbia, the welfare of sheep is little known, like in all Balkan region countries. So farmers and people, the general perception of sheep in extensive system living nature lives with few welfare compromises has caused them to be largely ignored in comparison to other species. So the sheep in extensive environments may face a range of compromises to their welfare, but principally this relates to variation in climate, especially in high temperature, since low temperature more affect the lambs than adequate or suitable nutrition, attack of predators, of course, a lot of parasitosis, since extensively reared sheep grazing on pasture, who is the most have a lot of parasitosis, and on the end, of course, human-animal relationship is sporadic and seasonal in this reared sheep. So aim of our study was to identify the main welfare issue during winter with an emphasis on parasitologic infection in extensive sheep farming system. Like the first speaker says, we want to see, we all know that parasitosis cause a lot of health problems in animals, but here we want to see which welfare parameters causes from parasitosis. What we done, this study we conducted during the winter season of 2022 in Serbia, in mountain region of Fruška Gora, in 3 farms, and that farms include native breed of sheep, Vlašićka Zakel, and this kind of sheep all year round graze in paddock where the water is provided from natural spring, and shelter from varying climatic extremes is provided by trees, shrubs, and other vegetation. No artificial shelter was provided for ewes or for lambs. And these 3 farms included 550 ewes, 12 rams, and 300 lambs. So we decided, according to AVIN protocol, to obtain 82 ewes aged 2 to 7 years old. For welfare indicators, we use AVIN welfare assessment protocol for sheep. We only use animal-based measures like body condition, fleece cleanliness, lesions, respiratory diseases, post-qualitative behavior assessment on the end. For parasitologic examination, we done coprological examination, macroscopic and microscopic from feces, from examinated ewes. We done qualitative VIDA method, flotation and sedimentation techniques, and quantitative McMaster method. And on the end, we done Spearman correlation between animal basic measures, some parameters, and examined parasites. What results show? Firstly, I want to show you what kind of parasites we found. In all sheep, we found co-infections with a lot of parasites, and this picture you will see, it's our picture we took from examined feces. Firstly, we found 95.10% prevalence of coccidia, then 92.7% prevalence of strongyloid, then 58.50% prevalence of the Fusarium dendriticum, then 34.20% prevalence of protostrongyloid, then 34.20% Magnesia, then 22% Buxtonella sulcata, and in the end 9.76 prevalence of Trichuris ovis. On this slide, we can see that the coccidia strongylide was the most prevalent parasitosis we found in examined sheep, and only coccidia was with medium and high intensity. Another endoparasite was with low intensity. That means that these endoparasites show only in subclinical forms, and that can affect economical part of sheep farming, and of course, like we will see after, and affect animal welfare also. Regarding to co-infections, we found double, triple, quadruple, fivefold, and sixfold co-infection. Most prevalent was triple co-infection with Coccidia strongylide and Protostrongylide, and Coccidia strongylide Epithelium dendriticum. And this polyparasitism, which we found in examined sheep, can influence the host immune system and increase theirs to other diseases or parasitosis. And we can, this parasitism, polyparasitism, might be due to sheep grazing on contaminated pasture, because farmers doesn't do rotation of the pasture, or unsystematic deworming or not treating, because these farmers treated their animals 1 or 2 times during the year, on autumn and the spring, but only ivermectin. It's not possible to affect all endoparasites. So our animal-based measures, what we found, that this presentation will be better with slideshow, but never mind. We found the thin body condition score in 15.85% of the eaves, and they significantly correlate with fleece quality. Even if we didn't found the correlation with any parasites, but we might link the high prevalence of coccidia with these results, because coccidia otists influence the damage of intestinal cells. So animals can't absorb nutrition or other minerals of vitamins. Other animal-based measures, animal indicators, is fleece cleanliness, and fleece cleanliness can provide us information about weather. And of course, we got 84.40% of dirty fleece cleanliness animals, and this result is related with extensively managed reared sheep, weather, and no provided shelter and bedding. Then lesion on legs, we found a low lesion of 4.88%, and this can be related with activity of eaves to grazing, foraging for nutrition. And when nutrition is low, resource is low, they forage through the scrubs, vegetation, and can get lesions. And what we found was interesting with correlation, that nasal discharge significantly correlated with Protostrongylide. Protostrongylide are the lungworms. So lungworms cause the pyrexia, coughing, shallow breathing, and nasal discharge. So this was the really problem, because of 45 0.12 EVs show nasal discharge. Borderline anemia significantly correlated with Buxtonella sulcata, also with endoparasite protozoa. And on the end, we found the minor lateness. Even was the low number of animals with this condition, but it's generally known that this is one of the main welfare problems because it's painful, and a lot authors talk about these problems, especially in extensively reared animals, because farmers, it's hard to find animals with these problems. So these animals can suffer weeks or months until some help or treatment. Like we see here, and we know also a lot of problems occur in extensively reared, especially native breeds. Qualitative behavioral analyses show that even their health is influenced by parasites or some other causes. Positive emotion is most expressed regarding to negative emotions. So what we can do, especially in developed countries such as Serbia, we need a well-coordinated sanitary monitoring of sheep farms by field veterinarians. But firstly, we need to improve local infrastructure because our infrastructure is so bad. So can pass weeks or months until any veterinarians came to field to treat the animals. Then provide adequate physical living condition, example, artificial shelter, and of course, on the end, dissemination of knowledge to animal handlers and farmers, and not only for them, and for some competitive authorities to improve animal welfare, improve occurrence, maintenance, and speed of parasitosis. So thank you for your attention, and I'm apologize because I really didn't found a way to come and be part.
Thank you very much for your presentation. Dealing with all these challenges, we have time for one question, one brief question, brief answer. Yes, please, here in the middle again. There's the microphone coming here in front.
Hello, Katerina. What a really lovely talk you gave there and a really thoughtful analysis. This is Suzanne Millman from Iowa State University. I believe you only measured the animals once, so you would not be able to determine the burden of the parasites before some of the welfare indicators. But I wondered, I saw in your abstract, you had quite an range of ages. And I'd be interested to know if you had looked at the data in that way, particularly with the use.
Did you understand the question properly? The question was mainly, did you look also at the age groups? And also, I think maybe, did you? Or maybe you can't hear us anymore, Katarina?
Hello? Hello? Hello, I'm here. Can you hear me?
Yes, we can hear you. Yes, yeah.
Okay, share something, some doing, and I don't know what. Okay, I hear your question and thank you. This is a part of biggest project, so we will try to spread our results. We didn't do. We done only 2 to 7 years old animals, but will be interesting to done even lambs or older animals. I don't know if I understand the question because something was wrong with.
Oh, I think it's okay. Thank you very much for the answer. And thank you for staying with us. And yes, we will move on now.
Okay, thank you.
I would like to welcome our next speaker, which will be again with us. And we are now changing the species. We're moving to laying hens. And I welcome Katalin Wurz from Denmark. And she's talking about the Association of Range Use, Individual Behavior and Welfare Indicators of two laying hen hybrid housed under organic conditions. Please, go ahead.
Thank you for the introduction and thank you all for being here today. So free range systems for laying hens are growing in popularity, in part due to consumer demand for more welfare-friendly products. These systems do allow hens to express a greater number of natural behaviors. They also provide individuals with an increased diversity of foraging materials, and they can also effectively increase the amount of space available per individual bird. However, there is quite a bit of variability in how much of the flock actually utilizes the range. And this can be due to a number of factors such as age, the genetics, their rearing environment, or even the weather. However, even within a single flock housed under the exact same conditions, we do observe a bit of individual variability in how hens use the range. So that's both how often they access the range and also how long they stay on the range. Although the actual welfare of these different range use personalities, I guess, that we have is poorly understood. So therefore, the objective of this study was to assess if the extent of range use at the level of the individual was associated with several clinical welfare indicators and tie budgets. So there is the possibility that the increased choices that a range provides a hen could impact their condition. Conversely, it's possible that a hen's condition could impact her choices and use of the range. So our study birds came from a population of 1,200 non-beak trimmed laying hens housed under organic conditions. Half of them were decal white and the other half were Bovins brown. And they were housed across 12 pens containing 100 birds each. And these were all single strain pens. From each of these pens, 10 individuals were randomly selected to serve as our focal birds. I mean, over here, you can see a photo on the right of what the housing looked like indoors. So the hens had access to perches, water lines, feed pans, nest boxes, and the floor was also covered with wood shavings for litter. Each of these pens also had access to a range area for each of the flocks. The birds could access this through a pop hole that was opened every day at 8 in the morning and closed again 45 minutes after sunset. And they were able to access the range beginning from the day that they arrived on the study. You can see in this photo here what the range actually looked like. So at the start of the study, it was covered in vegetation, which was mostly grasses. And then there were shades provided. They were spaced across the entire length of the range. So we performed a welfare assessment on each of these 120 individuals at 5 time points throughout the study. So we examined them at 18 weeks, which was when they arrived at our facility, 23, 28, 33, and 38 weeks of age, which was the end of our study. The welfare indicators we chose to assess were plumage condition of the breast, wings, and tail. And the plumage condition was scored using a 5-point scale, but for the analysis, we actually dichotomized it into either great or good, feather cover, or moderate or severe damage. We also assessed keel bone fractures and deviations. We looked for their presence or absence using the palpation method. And if they were observed, we noted what location of the keel bone they were at. We also assessed the individual's condition of their feet. So looked for presence or absence of bumblefoot, hyperkeratosis, or lesions. And then at each of these time points, we also took each individual's body weight. And during this handling, this is when each of our focal birds were given a backpack with an individual identifier on them. You can see a photo of the backpack there. So for the behavioral time budgets, we assessed behavior through direct observations. And we did this over 5 3-day periods that corresponded with the same weeks that we performed the welfare assessments. So each of the ranges were scanned, and then each of the indoor pens were scanned. And this was repeated a total of 6 times. And this occurred from 8:30 in the morning until 15:30. And the behaviors that were recorded included resting, perching, standing, locomotion, comfort behavior, dust bathing, foraging, eating, and drinking. On range use, this was assessed through the use of cameras that were mounted above each of the pop holes. And video was recorded, and this was observed for 3 days at 5 time points that corresponded with the time that we were performing the behavioral observations. From these observations, we were able to calculate the duration of time that each individual spent on the range and the number of exits that they took through the pop hole to get to the range. And these values were averaged across the 3 days for analysis. So quite a few models were fit. I'll just give a brief summary of what we did. For the binary responses, we fit mixed effects logistic regression models. For the ordinal variables, we use proportional odds models. And then for the counts of behavioral observations, we fit Poisson models. For all models, we included fixed effects of weak, hybrid, and range use, and their interactions were significant, and the random effects of pen and hen ID. And the response variables were each of the clinical welfare indicators, as well as the counts of the behaviors. So here I've got some descriptive statistics for the duration of time that hens spent on the range. These are in minutes and for each broken down by each of the weeks that we observed hens. So all of the hens in our study did access the range at each of the days that we observed them. I just want to draw your attention to the minimum and maximum values that we observed individuals ranging. The minimum was 0.3 minutes in a day, and the maximum was 710 minutes in a day, and that equates to about 12 hours of time that they spent on the range in a single day. On the right here, I have a histogram showing the variation within the population. You can see that most hens ranged between 0 and 500 minutes per day. It is much more unlikely to observe hens for greater than 600 minutes per day. I have a similar table here for number of exits onto the range for each of the weeks. And we see that the minimum number of exits that a bird took was 1 in a day, and the maximum was 106. And that equates to roughly, I think, 7 exits per hour for the duration that they had access to the range. And then again, a histogram displaying how the individuals were exiting the house. And you can see that one individual seemed to be a rarity that exited so often. Moving on to results, neither the time spent on the range nor the number of exits to the range were found to be associated with the clinical welfare indicators of tail, wing, and breast plumage, keel fractures or deviations in any location, bumblefoot or hyperkeratosis, or body weight. However, we did find a negative association between number of exits and foot pad lesions, meaning that individuals with foot pad lesions were less likely to be observed exiting the house. And while we can't infer the direction of this relationship, one possible explanation could be that these individuals with lesions were maybe in pain and maybe were not as active as other individuals, and thus would be less likely to be observed exiting onto the range. Moving on to the results for the behavioral observations. A duration of time on the range was negatively associated with the behaviors of drinking, eating, nesting, and perching. And this makes sense just based upon, if you think about where the resources for these individuals are located, the drinkers, feeders, nests, and perches were all located within the house. So if they're spending more time out on the range, they are probably less likely to be observed engaging in these behaviors. We did find positive associations with time on range with the behaviors of foraging, dust bathing, and standing behavior. And then we looked at number of exits, we found negative associations with exits and perching behavior and resting behavior, and positive associations with locomotion. So here I just want to highlight, I think these are pretty interesting results, that it seems that individuals that were spending more time on the range or exiting to the range more often seem to be more active, engaged in more locomotive behaviors, and also engaged more in foraging and dust bathing behaviors. In summary, the extent of range use did not appear to be associated with the clinical welfare indicators that we observed, apart from footpad lesions and number of exits. Behavioral time budgets differed depending on where they spent their time and what resources were available to them. And those individuals that used the range more were more active and engaged more in natural behaviors. So in conclusion, the lack of observed associations between welfare indicators and range use may suggest that hens were able to self-regulate and adjust their behavior to cope with their environment. Further, by encouraging range use, we may also be able to potentially encourage birds to be more active and to engage in more natural behaviors such as dust bathing and foraging. And then finally, I think that the provision of environmental options may help improve welfare at the level of the individual, as opposed to providing environments that cater to the average hen. And finally, I'd like to thank our sponsors. This was part of the Free Birds project that was funded by the Core Organic Co-Fund, and then we received additional funding from the Innovation Fund Denmark. So thank you for your attendance. I'll take any questions.
Thank you very much for this clear presentation. And we'll have many questions. We start here in the front, then go behind you, and then go to the other side.
Hi, Saskia Kliphuis, Utrecht University. Thank you for your talk. In your conclusions, you mentioned that it's good to motivate the birds, to encourage the birds to use the free range for all the reasons you gave. Do you have any suggestions how to encourage the birds to use the free range?
Yes, so one big thing is to design ranges that birds feel safe to access. This can be by including maybe trees or cover, bushes, vegetation, things to actually draw them outside. I know providing more shade and cover also can help pull them out further into the range. Yeah, I think that's really just giving them an environment that they feel comfortable in actually accessing. And also, I think another big thing that you can do is give them earlier exposure. So these birds in my study actually got access to the range during the rearing period, which I think is pretty rare. I think that can definitely help encourage birds later in life to actually get out and use the range. Yeah.
Yes, just behind. Yeah.
Sabine Gebhardt from University of Bern. Yeah, thank you for the data. You lumped the data over days, right? Did you also look at the variation among days? And because you lose a lot of information, so did you make sure that there was nothing in it? So were they really constant during successive days?
So within the 3-day block that we looked at?
Yeah, yes.
Yes, they were pretty similar. We did observe, I think it was the coefficient of variation between those days. And then also we looked at hens across the entire time they were on our study. And it seemed like individuals were fairly consistent in how they were using the range on a daily basis.
Okay, thank you. So you didn't have any outliers, gross outliers?
There were a few that I think would be interesting to follow up on in more detail, but most of them were fairly consistent.
Okay, thank you.
Yeah.
Yes, and now just beside you.
Mike Toscano, University of Bern. Interesting study. I was confused about one of your conclusions, where you suggested that the evidence suggests that animals are self-regulating, but you didn't find a correlation between the behavior and between the behaviors and the welfare indicators. So it's just a lack of relationship. So it would almost suggest they're not self-regulating. So that they're not adjusting their behavior, even though they have keel bone fractures. So could you explain that? Do I have that right, or am I missing something?
I guess my reasoning behind was they were able to, it shows that they were able to cope. So if they were experiencing something that would potentially lead to a negative welfare indicator outcome, they were able to avoid that. So maybe like feather pecking, they were able to move to the environment that it was best suited for them. I think I interpret it as an ability to cope, and I don't know, I might be wrong.
Yeah, there was one more, I think, in the back. Yes, behind you. Then that's the last, maybe in between the people presenting posters in person, start to get ready.
Thank you very much, Clara Gitten, University of Bern. My question relates with the variation between individuals you had. Could you just elaborate a little more how different the hens were in their outdoor range use?
Yeah, I could go back actually. So yeah, here looking at this histogram, I guess that's probably the best visual representation I have in individual diversity.
So it seems like you barely had hens that didn't go outside at all.
Right, all of the hens did access. If you look at the time, like there was this first bar graph here on the left, there were quite a few that only went out between 0 and 20 minutes. So it's few, but it was pretty evenly distributed across the time. Yeah.
Okay. So thank you very much for your nice presentation.
Thank you.
The next presentation, which was supposed to be, I think, is now not. Or is the person here? The last in this session would have been kind of a change of continent to Ethiopia, animal welfare and changing agricultural landscapes of Ethiopia. But once more, just asking if Gesa Gain Aylaf is here, but I think it's not online. So no, so we are sorry not to have this presentation. However, we are looking forward now to the poster session. And yes, as announced, we will start with the first 4, which are in-person presentations. And please line up and remember that it's only 1 minute short, but it's not the quantity, but the quality. So we will start with Lucy Markland, please, which is already here. Yes. Please, go ahead.
Hi, everyone. My name is Lucy Markland, and this study investigated the effects of jute nesting material and enriched piglet mats on sow and piglet welfare. So we had this idea because sows, as we know, are really highly motivated to perform nest building behavior prior to farrowing. We also know that when they're able to do so, there's this cascade of beneficial effects for not only the sow, but her offspring as well. However, conventional systems such as the barren farrowing crates that the majority of sows in the United States are kept in, restrict this behavior pretty severely. So we don't get to see this cascade of beneficial effects. Therefore, the aim of this study was to test the effect of an alternative nesting environment on sow and piglet welfare. So we did this by using 20 sows, half were the treatment group. They were given jute nesting material attached to the side of the crate. They were also given thick microfiber piglet mats to mimic a nesting environment for their offspring. And then the control group were housed just in a traditional barren farrowing crate. And we did see some interesting positive effects for both the sows and piglets. And I'd love to go into more detail about those because we don't have time today, and the implications of this study later today at the poster session. Thank you.
Thank you. Perfect. And the next one is Lene Juhl Petersen.
Yes, my name is Lene Juhl Petersen. I'm from Aarhus University in Denmark. And the results that we present in this poster is part of the EU project, the P-Pillow. It's a project where we investigate features to improve welfare in sows that are kept outdoor on paddock with access to a farrowing hut. And we know that when sows are kept outdoor, which is obligatory in Denmark for the organic production sows, then we facilitate much more positive welfare. For example, they can forage freely, they can move freely around, and they can nest build and show all their maternal behaviors. But the problem is that we see quite a high piglet mortality in this system. So currently in Denmark, it's close to 30% of the total born piglets. So therefore we need to work to improve that. And that's what we are doing here. And our approach is to look into the design of the hut to see at how different features inside the hut affects the possibility for reduced piglet mortality and also improve the growth of the piglets. So please come and hear more about the results at this poster. Thank you.
Thank you very much. And the next one is Marc Bagaria, and also about piglets.
Yeah. Hello, my name is Marc Bagaria. I'm coming from Mirta in Spain, and I'm gonna present this poster about nutritional strategies to prevent post-weaning diarrhea in post-weaning in organic piglets. The main objective of this study was to assess if a decreased crude protein diet or a decreased crude protein diet supplemented with liquid whey, which is a byproduct of the cheese industry, would prevent post-weaning diarrhea in organic piglets, and without compromising their behavior, welfare, or growth rate. The main conclusions of this study shows that neither both of the diets decrease post-weaning diarrhea, but that the diet of a low crude protein diet supplemented with liquid whey increased daily weight gain, which indicates that liquid whey could be an optimal supplement for organic diets. And if you are interested in knowing more, in room number 2, poster 70. Thank you.
Thank you very much. And the next one, I think there's one online. Is Gudrun Illmann here? No, I think she's online. Gudrun? She here? Number 71. Confinement for 3 days postpartum. Otherwise, we have one more in person here, as far as I can tell. We'll check for her. We will check for her. Yeah. Otherwise, we move on in between. Is this okay? She's not here. Okay, so we move on to Mirjana Stojcic. Mirjana, please.
Thank you. I'm Mirjana Đukić-Stojcic. I'm coming from University of Novi Sad, Serbia. In this study, actually, we investigated the effect of production system, of behavior, welfare parameters, production parameters, and as well meat quality of middle growing broilers. In this, for this purpose, we use red brow, middle growing chicken, that we raised on our experimental farm 8 weeks. And during the 8 weeks, once a weekly, we observed the birds as well. We took the welfare parameters, we showed the welfare parameters, and on the end, we slaughtered the birds and showed the meat quality and calculated production parameters. In this paper, we showed us the results of our observation, and more results you can see on our poster presented as 72 number. And the main conclusion are that production system had an effect on behavior and welfare of those middle growing broilers. Thank you.
Thank you very much. Now we move to the sheep, number 73, online. Yes. Please go ahead.
Hello, my name is Madeleine Guerrero. I'm from Uruguay. I am a PhD student. The object of this experiment was to determine the basal behavior during the days after shearing, time and distribution of time consuming food, rumination, standing, lying down, and walking. 22 other choreal rams were assigned to 2 homogeneous groups, while 11 rams were sheared, 11 remains a control. Rams' behavior was recorded 3 days before shearing and 7 days after shearing. As a result, sheep were observed standing and walking more frequently than, but less frequently lying down than woolly rams. In conclusion, after winter shearing, rams increased their physical activity and decreased their time lying down, probably to save energy since it generates heat and decrease heat loss due to wool removal. Thank you.
Thank you very much. And Gustavo Diaz, number 74, online. Are you here? Please share your screen. No, not here. Let's see.
Yes, I'm here.
Yes, perfect.
Sorry. Share screen. 1 second, please. Can I go after? Then I'm going to be ready with my screen. Sorry.
Okay, so we move first to 75. Caroline de Souza. Is she here? No, not online. A video, hold on. So we do the video first. Is this okay for you?
A PhD candidate in Brazil. And my thesis, I am studying the effects of different tree arrangements of a silvopastoral system on cattle behavior. I perform a systematic review to summarize the data available in the literature on this subject. After the search and the screening of the resulting articles, we found 12 articles, 6 of which evaluated cattle performance and 6 evaluated cattle behavior. Most of the studies were performed in Brazil, and the main difference among the tree arrangements evaluated was the density of the trees. An interesting result is that cattle spent more time ruminating in higher tree density arrangements. Unfortunately, cattle behavior is scarce explored in the studies, and when it is evaluated, the authors did not use the same measure of the unit to present the results. Thus, it is difficult to reach a consensus on the effects of tree arrangements on cattle behavior. Thank you.
Thank you very much. Is Gustavo Diaz already ready?
Yes, I'm ready now.
Perfect.
Thank you.
Go ahead with your presentation. 1 minute.
Can you see it now?
Not yet. It's still black. Yeah. 1 minute. Maybe use the other screen or something. Yeah, here it is. Perfect.
Here it is. So the study is the influence of the bedding material on dairy cows' behavior. So we aim to compare the behavior of hosting cows housed with 2 different bedding materials, sawdust and sandy. So we housed 8 dairy hosting 80 hosting cows in 2 groups with sand bedding material and sawdust bedding materials, and we collect the behavior of those dairy cows in 6 days, and we measure the behavior of laying, standing, feeding, and ruminating, and the behavior of those cows differ, and the time those cows stood up,
on the grazing behavior of 32
and the
square design with 4 groups of 8
Sorry, we
animals
seem to have a
each.
double
We found
conference
out
here.
that juvenile
No?
grazing was higher for 4 hours water treatments.
We somehow
We
have
also found
someone else talking here. Okay, it's better now. So you can finish, I think, now with your talk.
Okay, thank you very much. So, so we measured and we recorded the behavior of every 5 minutes of those cows, and as a result, the times that those cows stood up differed, and the time didn't influence of any variable related with laying, and the cows in the same treatment had shorter meals than the cows in the sawdust, and the cows in the sand had shorter meal time of rumination, and this sand cows had more events of rumination per days, with and those events dured less time. So the conclusions was that although the behavior of those cows differed, the cows showed a normal daily budget in both bedding types. So those both bedding materials can be recommended for dairy cows. Thank you.
Thank you very much. So we are almost there. There's a number 76, another Luis Carlos Pinheiro.
Areas. The availability of shade and water are of paramount importance for the behavior and the welfare of cows. We investigated the effect of water and shade availability on the grazing behavior of 32 cows in a 4x4 Latin square design with 4 groups of 8 animals each. We found out that diurnal grazing was higher for 24 hours water treatments. We also found that water restricted increased diurnal idling, and no shade decreased diurnal laying time. Apparently, water availability is more important than shade for diurnal grazing. When no shade is available, they lay less, probably because the ground temperature is high, and standing facilitate the airflow over their body. When water and shade were available, of course, cows had a higher diurnal grazing and
this was now a bit abrupt, but thank you very much. And we move on. Yeah, that's good. But we move on now to the last two ones, exciting species. Red panda, please. Where's the red panda? We need to change the screen again, back to where we were before, please. The red panda was already there. Let's see, is Ayuka Sanada here? Yes, this is the poster. But online, yes, as you can see, Ayuka Sanada, just switch on your microphone, maybe. No, try again. Yes, here. Let's see if it works.
Yes, can you hear my voice?
Yes, perfect, now you can start. Go ahead, please.
Environmental temperature is one of the factors that have an important influence on behavior of animals. The captive animals are often bred in a climate different from their original habitat. In animals living in alpine areas, it is necessary to be careful in the hot summer condition. In this study, we investigated the effect of hot summer condition on behavior of captive red panda. There was a significant positive correlation between the temperature in the exhibit room and the spent time in the coolback room. In the exhibit room, there was a significant negative correlation between the temperature and the spent time. These results suggested that the red panda may have moved to the coolback room to avoid hot. Thank you.
Thank you very much. And now, last but not the least, my favorite species, working donkeys. Let's see if they're there. Just try to find the presentation. Are you there? Syed Saad Ul Hassan Bukhari? No, that's the wrong slide. Yes, here we are.
It's not online and there's no video material.
Okay, so we can just look at this and think of all the donkeys working in all different parts of the world, acknowledging their work. And I acknowledge your presence here. I think it has been a very nice session, and we are all looking forward now to have some lunch to care about our welfare. I'm not sure if there's now this announcement for the afternoon working groups. Is it or not? No, they're not here yet, Vlatko. Maybe we will do that after lunch straight away. So thank you very much. And yes, it was a lovely session.